Mhm. Good evening, Vanshika. Okay, so I'm Rahul Saha, and today we'll be talking about more about the cleavage that we have talked about, the compaction and the types of cleavage, and we'll also be talking about the implantation. Okay, how does the implantation take place? So those things we'll be talking about. So let's begin with more of the specificities regarding the cleavage division. Okay, so when we talk about cleavage, okay, so I told you last time that cleavage forms the cells that are known as blastomeres. Okay, and in cleavage, if you talk about cleavage mitotically, it gives rise to blastomeres. This is a very important question. If you look at cleavage from mitotic standpoint, okay, mitotically, then it's a very, you can say, amazing event that the cell divisions are very rapid. Okay, and the cell divisions are so rapid because you need to produce, you need to divide the cells at a rapid rate and also need proteins at rapid rate. So the cell cycle is quite rapid, and in order to make it rapid, the G1 and G2 phases of the cell cycle are completely eliminated. The cell cycle only consists of, you can say, the S phase. Here only organelle duplication, DNA duplication will occur, and then the M phase. So G1, S, G2, M, so only S and M phase, they remain in the cell cycle. Okay, in the mitotic cell cycle. There's a difference between a mitotic cell cycle and the cell cycle pertaining to the cleavage. Okay, next is the cleavage, like with the increase in the number of cells, what happens is the cells become smaller with time. Okay, cells become smaller with time because we have more and more cells surrounded by the zona pellucida, so they become smaller with time. So what you have is the first cleavage division. If you talk about cleavage division 1, 2, 3, 4, so when you talk about the first cleavage division, in your first cleavage division, what you have is you have the resulting embryo, which is of two cells, because on the first day I told you will get two cells or two blastomeres, and DNA has been synthesized. Now embryo has double the DNA content of the zygote. Okay, and you can say no real growth of the entire cell has occurred. Okay, so the two blastomeres together are approximately of the same size as the zygote, means the zygote growth has not occurred here. Okay, so what happens, the blastomeres, they keep on increasing. The length or diameter of the zygote, it stays the same, one cell, two cell, the growth stays the same. So it doesn't grow, this one doesn't grow to accommodate. Okay, you then the cells will become smaller. Second cell division or second cleavage, if you see, here also what happens is the growth has not happened, but you have now four cells. Okay, Now the membranes have been produced in the two cell stage only to surround two cells. Here it will be surrounding the four cells. Okay, but growth again has not occurred. Growth has not occurred. Okay, that is the problem. Now you have four blastomeres which are about the same size as the original zygote, right? Four blastomeres are the same size as the original zygote. So that is your second cleavage division. Third cleavage division, again embryo has 8 cells now. Okay, now embryo has got 8 cells, still growth has not occurred. Okay, still growth has not occurred. Third cleavage, 8 cells, still no growth. Okay, that's the dilemma of the situation. Now again, you will see the zygote is of the same length or same diameter as the 8 cell stage. Now the thing that I have taught you, compaction will come into action. Okay, third day compaction. Okay, this is like 2.5 days. Okay. Now after 3 days you will have compaction. Now you will be having, you can say embryo will undergo compaction, E-cadherin expression that we had already talked about, right? He will undergo compaction, and this will lead to the formation of lead to the formation of the 16 cell stage known as the morula. Okay. Now morula will be produced. After that we will be having, you can say blastocyst formation, then hatching, then implantation. So at 8 cell stage we know that compaction occurs. Okay, adherence occurs, E-cadherin expression occurs. Now after those things have happened already, okay, we know that E-cadherin is a calcium dependent cell adhesion molecule. So after those things have happened, we will go towards the formation of blastocyst. Okay, now when we have produced blastocyst, what did I tell you regarding that? It will lead to cavitation. Okay, begins 4 days after fertilization. Always remember the date. Morula 3 days, blastocyst 4 days after fertilization, right? Now in blastocyst we will be having cavitation, then or known as blastocyst cavity. So cavitation process, okay, influx of Na plus and water, we will be filling it with blastocele, water cavity. Okay, now once these things have occurred, okay, then we will shift our attention towards hatching and implantation. Okay, assisted reproductive technology is there, so those things may be used, okay, by the scientists for the same purpose as RT, you can say in vitro fertilization and all. So those things can take place. Now if you see the development of the first 35 days, I'll just give you an overview. Okay, so days 0 to 7, if you see, so this is a good table that you can think of. Okay, development, the first 35 days. So gastrulation is not in your course at all, so we won't be dealing with gastrulation, but other things I'll be explaining. So days 0 to 7, what you have is fertilization. Okay, then we have cleavage, then blastocyst formation. Okay. Then days 8 to 14, we have implantation. Okay. Onto the endometrial lining. Then days 15 to 21, okay, we have gastrulation, then vascular system development. Okay. The process of gastrulation occurs. Okay, I'll tell you what gastrulation is in very short. Okay, because those things are not there in your course. Okay, so gastrulation is an event. Okay, in developmental biology, gastrulation is a phase early in the developmental or early in the embryonic development of most animals, during which the after blastocyst it is known as blastula. So the blastula is reorganized, okay, into multi-layered structure known as gastrula. Now gastrula will contain what? Gastrula contains nothing but you might have heard in class 11th as three embryonic germ layers. Okay, ectoderm, mesoderm, and endoderm. Right? Now this gives rise to the nervous system, skin and all. This gives rise to the heart, muscle, connective tissue. This gives rise to the internal structures, right? Pancreas, liver, urinary system, reproductive tract. So unless this gastrulation happens, you wouldn't be getting proper morphogenesis. Morphogenesis means development of various organs. So morphogenesis would not happen. Morphological development, metamorphosis, okay, that we talk about in technical terms will not happen. So if gastrulation is problematic, then you would develop some or the other, you can say, enormous congenital defect in maybe the hole in the heart, or you can say three legs, two legs, okay, we can say conjoined legs, and all those things can occur, right? So there was a, or there is a scientist known as a great embryologist named by the name as Walpert. Okay, so that scientist had quoted. Okay, I'll tell you his quote. It's a very interesting quote, and all the biologists should know it. So one of my, you can say, favorite quotes with respect to biology, that it's not marriage, life or death that's most important. The most important event in the life of a human is gastrulation. Okay, that's the biological significance of gastrulation. Like if the gastrulation goes wayward, you have like twisted head, this and that, 100 defects. Your entire birth will be useless, right? Nobody will marry you, and your death will also be painful. So if your gastrulation is not appropriate molecularly or from the molecular basis, then the entire life will suffer throughout, even if you manage to live. So the thing is, the gastrulation is the most important event in a person's life biologically. Okay, so that is the meaning of gastrulation. Okay, after gastrulation, you have days 22 to 28. You have something known as neurulation. Okay, just I'm telling them the name so that you know the perspective of the human development. You get the broader picture because we are not going into details of gastrulation or neurulation. I'm just showing you the perspective that you should know that how do we develop as a complete baby or fetus. Okay, neurulation is nothing but neural crest development, migration of the neural crest, embryonic folds, development of lungs, urogenital system, the heart starts to develop, okay, arm, limb, bud appears, ears start to develop, okay, and then days 28 to 35, you have something known as nervous system development, okay, nervous system development. Then you can have the development of eyes start to develop, okay, then leg, limb, bud development, spinal column and ribs they begin to form, okay, peripheral nervous system develops, stomach develops, okay, then the external genitalia begin to develop as male or female, means at that point in time you get the identity that it could be a male or female, you start to get the sexual demarcation, then only at days 20 to 25 it starts, it commences, right? So this is the overall idea of how does a person or how does an embryo, okay, how does embryogenesis occur, okay, in any, you can say, human being. So that is the idea behind that. So now coming to our next topic, I'll tell you a little bit about, okay, a specific thing about cleavage. Okay, cleavage could be of two types. That is asked in NEET examination. One is holoblastic. Also, this is given in your reference book. If you open ABC reference book or any Pradeep or any standard reference book, that is given. So I'll tell you that in detail because in ABC they have made some mistakes. Okay, so I'll just clarify the mistakes for you and just follow the classification that I am providing. That is error free. So types of cleavage, if you see, okay, types of cleavage, this is asked in NEET examination directly. You have something known as holoblastic cleavage, known as complete cleavage. Okay, holoblastic cleavage, that is the complete cleavage. Then you have something known as, you can say, meroblastic cleavage, which is known as incomplete cleavage. Meroblastic or incomplete. Incomplete cleavage. Now under, if you check holoblastic cleavage, okay, so I'll show you an image, okay, that image will be there in this recording also, so you can watch this recording again. So under holoblastic cleavage, you have various types of yolk distribution in the egg. You have isoleucithal, where evenly distributed yolk is there, okay. Then you have something known as mesoleucithal, where moderately or moderate distribution of vegetal yolk is there, okay, at the vegetal end, not the animal pole, the vegetal pole, trophoblast. Then meroblastic, when you come to, you have teloleucithal, dense yolk throughout most of the cell, okay. And then you have centroleucithal, okay, which is yolk in center of the egg. So those things you have. So the thing is, what we show is, we show the isoleucithal because we don't need too much yolk. We have the nutrients coming in from the placenta. So we have holoblastic cleavage, complete cleavage, and under that we have the, you can say, isoleucithal type yolk distribution. I'll just show the image to you to become very, very clear. Okay, just see the image. I'm sharing the screen. Okay. So now I think you are able to see the screen. You will be able to see the screen in 2 seconds. I think, yeah. I think the screen is visible to you now. Just look at this. Okay, image. This is holoblastic complete cleavage, isoleucithal. Okay, spares evenly distributed yolk. Okay, you can have radial cleavage, radius wise, spiral cleavage, like a spiral, bilateral. Okay, and there are written also which type of organisms. You have echinodermata, like starfish, and then you have annelids, mollusks here, flatworms here, spiral one, tunicates, bilateral, and rotational cleavage is present in mammals and nematodes. Okay, like this. This is how the cleavage happens in us. Okay, and it is isoleucithal because it is evenly distributed and spares very, very less yolk because we have placenta attached. Okay, so under holoblastic cleavage, you have an isoleucithal type and mesoleucithal type egg. Under isoleucithal, you can have 4 types. Okay, so these you don't need to remember. Just I'm showing it for your perspective. It will just come in the exam. Mammals fall under isoleucithal cleavage, holoblastic. Okay, radial and all you don't need to remember. And then we have mesoleucithal egg, which is moderate vegetal. At the vegetal pole, it will be distributed, okay, in the amphibians. Then we have meroblastic cleavage. Under meroblastic we have telolacethal, okay. Then bilateral and discoidal is there, okay, which is dense yolk because they lay egg, right? And centrolacethal, again dense yolk, but it is at the center, most insects, okay. So this is the entire, you can say, phenomenon governing the holoblastic and meroblastic cleavage, okay, incomplete and complete. So you will have isolacethal yolk, you will have telolacethal, centrolacethal, okay, mesolacetal. So under holoblastic, iso and meso, this thing will come in NEET examination, not the sub parts. Holoblastic, iso, meso, okay. Meroblastic is centro and you can say telo. Centro you can see here, telo you can say here, okay. So telo and centro there, and iso and meso under holoblastic. So this much you should be knowing for NEET examination at least, okay. So then we'll move on to the next topic, which is known as hatching and all. Okay, so this we have done, okay. So now when we have seen how does the blastomere, it divides to form 8 and 16 cell embryo, okay. What happens is, how will you decide, okay, the polarization of the blastomere, meaning that how will you decide the polarization of blastomere. Okay, at the blastocyst stage, means how will you decide which cells will become, or which cells will take the lineage of inner cell mass, which cell will take the lineage of becoming the trophoblast cell. Okay, we know that the trophoblast only produces extra embryonic structure, meaning the outer layers and middle layers of the placenta. Inner cell mass produces the intra-embryonic structures, means the entire embryo morphogenesis, metamorphosis, and also the innermost layer of the placenta, which is again an extra embryonic layer. So this forms both, as per the competitive exam, intra-embryonic as well as some extra embryonic entities, okay, or structures. This forms exclusively extra embryonic structures. Now, how does the cell decide upon that? Okay, what happens is there is a specific hypothesis that I like to call inside outside hypothesis. Very easy. Now, the essence of this is that the fate of a blastomere derives from its position within the embryo rather than its intrinsic properties, meaning that you will be having lots of blastomeres, right? At the blastocyst stage, you will be having one layer of blastomere, this one, another layer, this one, okay. So what will happen? How will you be able to, you can say, change your way or know which one to go? So the G1 and G2 stage that I told you, it goes off in cleavage, okay. When the blastocyst stage begins, okay, again the S phase, sorry, the G1 and G2 phase, they are restored. When the blastocyst begins, the G1 and G2 phases are restored. By that time, you have multiple layers of cells. So the outer layer of cell facing the zona pellucida would most probably become the trophoblast. That is the, you can say, inside outside hypothesis. The outside will become the trophoblast, the inside cells will become the inner cell mass. So one of the reasons is this one. If you think about more reason, okay, you can have a cell polarity model, okay. According to this, you see, if the plane of cell division of a blastomere at the 8 cell stage is parallel to the outer surface of the embryo, the daughter cell develops a polarity with its apical surface facing the zona pellucida, and the inner cell remains apolar and goes on to form the inner cell mass. Okay, so that is a little bit complicated one. So we'll skip that at your level. Okay, so that is another hypothesis. Okay, now the thing is, regardless of how it happens, okay, there are hypotheses like this. That's why I told how it happens. Regardless of what happens, even if you have reached the 16 cell stage, then you have reached the morula stage, means 16 cell stage. When you have reached the 16 cell stage, simply known as morula stage, okay, then we'll be having the parental imprinting, then we are which we have talked about. Okay, so at the 16 celled morula stage, that consists of polar outer cells and non-polar inner cells. Okay, the outer cells are known as polarized, they will become trophoblast. Inner cells are non-polar, they will become, you can say, inner cell mass. Now still either of these cell type can be transformed into the other cell. Okay, so meaning that the cells of the inner cell mass, if transplanted to the outer surface of another embryo, it can become trophoblast, and at least some of the outer cells can turn into inner cell mass if they are transplanted into the interior of inner embryo. Suppose here you have this one as the trophoblast, okay, and this one is the inner cell mass, non-polar, okay, and if you transplant this onto the outer side, these cells outer side of the embryo, these may become trophoblast, and if you transplant it in the inside, they may become inner cell mass. But only till 16 cell stage, that's a great amazing thing, because by 32 cell stage, this capability means many of the genes will be epigenetically silenced, and this capability for phenotypic transformation would be largely lost. Okay, so this type of phenotypic transformation ability, they have the ability that scientists have discovered, phenotypic transformation would be completely lost. Okay, now the thing is, we have many genes, okay, but the genes are not in your course. So in 2013, in case the Nobel Prize was awarded for induced pluripotent stem cell, okay, to Shinya Yamanaka and all. What they did was, their group was the first group to convert an adult cell into stem cell. That is what is known as induced pluripotent stem cell. It was induced, IPSC. Pluripotent stem cell was induced, means these cells were produced from adult cell. Just think. What they did was, they just again epigenetically expressed, okay, desilenced, you can say, many special genes like OCT4, NANOG, and SOX2. Okay, OCT4, NANOG, and SOX2. Okay, this is just for your information for the scientific purpose. Now these genes, they play a major role in developmental biology. Okay, that is not in your course, so we won't go down into the detail, but OCT4 is expressed in all blastomeres up to the morula stage. Okay, so those things are there, but we will just skip that discussion for now. And the thing is, we will now switch our discussion to, you can say, the specificities of, you can say this one, implantation. Okay, zona pellucida first and then implantation. Now during the entire period, so if we talk about the fate of zona pellucida, that is a great thing. Okay, that is a, you can say, enormous interest and intrigue both. So zona pellucida, it's such an enthralling, you can say, entity, acellular entity made up of proteoglycans, that during the entire period, it does contain receptors, zona protein ZP1 to ZP3 in mice, ZP1 to ZP4 in humans, right? So during the entire period from ovulation until entry into the uterine cavity, the ovum and the embryo, means the entire blastocyst, are surrounded by zona pellucida, right? So till you can say the embryo enters the uterus, the uterus, it is surrounded by zona pellucida. Okay. Now, when it is surrounded by zona pellucida, what is its significance? Okay. Now, what happens is during this time, the composition of the zona pellucida changes through contributions from the blastomere and the maternal reproductive tissue, both. Okay. Now, these changes facilitate the transport and differentiation of the embryo. Now, after the embryo reaches the uterine cavity, okay, it begins to shed the zona pellucida in preparation for implantation. So, after the embryo, again a great neat question. When does the zona pellucida shedding happens? That is when it reaches the embryo. Sorry, after the embryo, when the embryo reaches the uterus, right? So after the embryo, it reaches the uterus, it sheds the zona pellucida, or it sheds zona pellucida for in order to achieve implantation. Now, since implantation wants or is the need of the hour, otherwise the embryo will not be able to sustain itself, placental development will not occur. This process, okay, or this is accomplished by a process known as blastocyst hatching. Okay, the shedding of the zona pellucida is accomplished by a process known as, this milestone is accomplished by a process known as blastocyst hatching. Okay. Now, a small region of the zona pellucida. Okay, what happens is a small region of the zona pellucida, usually directly over the inner cell mass. Okay, in the primates like us and monkeys, dissolves and the blastocyst emerges from the hole. Okay, that is what happens. Now, what happens is during this time, okay, suppose we talk about the rodents because that's where the research has happened. The blastocyst hatching is accomplished, means you just hatch it like an egg, okay, because the inner cell mass and the blasts have come out, especially the inner cell mass. So what will happen is in primates, the first hole is produced directly over the inner cell mass region. Okay, the first hole is produced directly over the inner cell mass region. Okay. Now in rodents what happens is the blastocyst hatching is accomplished via the action of specific protease enzymes. Okay, 16 protease. You can just think of it as protease enzymes, protein degrading enzymes that are released from the microvilli or microvillus extensions protruding from the surface of the trophoblast cells. Okay, very great thing. So in rodents what happens is, if you go for blastocyst hatching, then the enzymes are the protease enzymes to degrade the proteoglycans. The protease enzymes are released by the trophoblast cells or by the microvilli of trophoblast cells, the microvillus extension of trophoblast cells. So that is what has been seen to happen in case of rodents. Okay. Now enzymatic activity around the entire trophoblast soon begins to dissolve the rest of the zona pellucida. Okay. So in humans, the only problem is nobody wants to participate in the research, okay, directly, because the baby's life would be in danger. So in vivo assays or in the body assays have happened a very less number of times, okay. So only a few specimens of human embryo have been taken in vivo from the period just preceding implantation, okay. But in vitro studies have happened, okay, that is there. So that is known as the blastocyst hatching, the dissolution of the zona pellucida, okay, the breaking or the dissolving or the breakdown of the zona pellucida in order to release the inner cell mass. That is the blastocyst hatching, okay. Now, when we come to implantation into the uterine lining, when does that happen? That is a great question. It will take 6 to 7 days in the NEAT examination after fertilization. So, implantation, when we talk about implantation, so occurs after 6 to 7 days of fertilization. That is there. Now, at this stage, what happens is the embryo begins to make a firm attachment to the epithelial lining of the endometrium. We have endometrium, myometrium, perimetrium. Endometrium is the most superficial line, sorry, the deepest lining of the uterus, okay, that is the cavity of the uterus. It surrounds the lumen of the uterus, right? So the endometrium is the deepest layer, and in implantation what happens is the embryo begins to make a firm attachment to make a firm attachment to the epithelial lining, to the epithelial lining of the endometrium. Okay, now what happens is soon thereafter it sinks into the endometrial stroma. Now stroma is the you can say connective tissue cells, just like we saw in the while, like during the development of the zona pellucida, sorry, the tertiary follicle or the Graafian follicle that I had explained to you about, the stromal cells of the ovary, they differentiate to become, you can say, theca interna and theca externa, which are, you can say, demarcated from the follicular cells or granulosa cells by the basement membrane known as the membrana granulosa, right? So here it sinks into the endometrial stroma, means the connective tissue just beneath the, you can say, epithelial lining of the endometrium, beneath the basal membrane, and its original site of penetration in the or into the endometrium is then closed by the epithelium, like similar to a healing of skin wound. That is what will happen. You can write it down and show you the image also. So its original site of penetration in the endometrium is closed into the, before that I should write about epithelial lining is done. Now here the, you can say, after this event it sinks into the, it sinks into the into the, you can say, endometrial stroma, that is the connective tissue, endometrial stroma, that is the connective tissue. Okay, connective tissue and its original site of penetration original site of penetration is, or into the endometrium, original site of penetration into the endometrium is completely closed off, is closed over by the epithelial lining, by the epithelial cell lining. I'll just show you the image, it will become crystal clear. Okay, just hold on, I'll show you the image, that how does it occur. Okay, so you might not have seen this image ever. You will be really surprised and excited at the same time after looking at this image. Okay, you will see this image now in just a few seconds. Yeah. Okay. So I'll share it. Just found the image. Just look at this image. Okay, you just look at this image. It will be a really, really exciting image. So this is how the entire perspective occurs. Okay, the entire thing happens. You can see here the laminin, the ECM is extracellular matrix. It contains collagen, fibronectin, okay, osteopontin, then you have trophinin, okay, SCG, various integrin. Integrin is alpha 5 beta 5 here, alpha 6 beta 4. There are various types of integrin, alpha 1 beta 3, okay. Then selectin, L selectin is there. Okay, so there are various types of cell adhesion molecules. So this integrin, selectin are all cell adhesion molecules, okay, that you are able to see in this. The main thing that I want you to know is, see that tropoectoderm here, okay, it is getting, the trophoblast is getting submerged and it is completely being, you can say, swallowed, engulfed by the stromal layer, okay. So that is what is happening. And then this epithelial layer, you can see the endometrial epithelium, the labeling the epithelial layer is just able to, you can say, coat it. That is what is happening. Epithelial layer is able to coat it, right? So this is what happens in the real time, you can say, or the reality of the event known as implantation. So this is what you really see happening during the implantation. Okay, so successful implantation, it requires a high degree of preparation and coordination by the embryo and the endometrium both. By the embryo and the endometrium both. Now the complex hormonal preparations of the endometrium that begin at the closure of the previous menstrual period are all aimed at providing a suitable cellular and nutritional environment, you can say, for the embryo. Now the first stage in implantation, it consists of or comprises of attachment of the expanded blastocyst to small surface projections of the endometrial epithelial cell, and those surface projections of the endometrial epithelial cells, I think it is drawn here, okay, they are known as pinapodes. This one you can see here, okay, these are the surface projections of the epithelial cell. To this they will attach in the beginning, okay, they are known as pinapodes, okay. Now during the implantation, the endometrium is covered with a layer of glycoprotein, okay. That glycoprotein's name is mucin, which is also present in mucus. Okay, glycoprotein means glycosylated protein. Glycosylation means attachment of glucose residue at N site, N linked or O linked, at the O site of the amino acid or the protein, N linked glycosylation or O linked happening at the Golgi apparatus via any glycosyl transferase enzyme. Okay, by that specific glycosyl transferase enzyme, and that mucin has protective and anti-adhesive property, okay, but the influence of blastocyst causes the down regulation of mucin in the area of attachment. So getting my point, the thing is, here if you see, mucin is present over the epithelial cell. What I'm saying is mucin is present over the epithelial cell, and mucin type 1 has anti-adhesive property, okay, and has also protective property. Now anti-adhesive means it won't let the adherence happen. Now when the blastocyst approaches, the signaling event leads to down regulation of production of mucin only at the area of contact, only at the area of contact, so that the anti-adhesiveness reduces, and then the blastocyst or can then successfully attach to the site of the epithelium or the epithelial lining of the endometrium where the pinapods are present. Okay, now the apical surface of the hormonally conditioned endometrium epithelial cells, okay, and the cells of the embryonic trophoblast, they express various adhesion molecules. Always keep in mind, this is the apical surface. Okay, this is the apical surface of this one, of this one. So they'll secrete various adhesion molecules, like you have L-selectin, you have integrins. Okay, various types of integrins are being secreted. So they all come under the cell adhesion molecules. Cell to cell adhesion, they are all transmembrane integral membrane proteins which will lock on with each other. Okay, and then they'll cause cell adhesion. Okay, by making the cell fix with another cell, or by fixing one cell with the other cell, by acting as a glue effect. Okay, it's known as cell adhesion molecules. So they do secrete, or they do express a lot of, you can say, various adhesion molecules, integrins, selectins, that allow implantation to occur. Okay, in the very narrow window, because only the implantation window is of how many hours? 20 to 24 hours in the ideal menstrual cycle. Okay, it is the only implantation window. Okay, in the ideal menstrual cycle, you have 20 to 24 hours, which are best regarding the hormonal level, which are considered best pertaining to the implantation. That is known as the implantation window. Now, the trophoblastic cells of the pre-implantation blastocyst, means before the blastocyst has been implanted, they also express, you can say, adhesion molecules on their surface, right? Now the blastocyst attaches to the endometrial epithelium through, you can say, mediation of this specific, you can say, adhesion molecules. So one adhesion molecule that you can see which is being expressed by the trophoblast, okay, is the L-selectin. Okay, and then you have another molecule that you can see here. Okay, that is known as troponin. These are all secreted, okay, on the surface of both the trophoblast and, you can say, the endometrial cells, which facilitates the adhesion. Okay, and also triggers the activation of the trophoblast, and which will give it the invasive power. Now the trophoblast will try to penetrate, you can see, will try to penetrate the epithelial layer of the endometrium and try to get into the stromal layer. It will try to get into the stromal layer. That is what it is trying to achieve here. Okay, it is trying to penetrate. It will provide the invasive property to the trophoblast cell. Okay, so many studies have shown that the attachment of blastocyst, okay, usually occurs at the area above the inner cell mass, means the embryonic pole. Okay, and what does this mean? This means that the surfaces of trophoblast, means the trophoblast cells, are all not the same. Means that what you see here is, this is the embryonic pole, this is the vegetal pole or embryonic pole, this is the animal pole, and this layer of trophoblast is attaching, which is at the animal pole. Means animal pole means what? The embryonic pole means what? Which is just surrounding, or which is just in contact with the inner cell mass. Why not the other way around? This could have also attached, but this is only attaching. So much reasoning is there in this, you can say, subject. Only this region is attaching, means I can say conclusively that only and only if this region is attaching, means these trophoblast cells could be little bit different in expression of various cell adhesion proteins in comparison to the trophoblast cells present at the embryonic stage, or sorry, at the embryonic pole or at the vegetal pole. So there could be a great difference. That is why always 100% of the time this only occurs, that the attachment of the trophoblast which is at the animal pole only occurs, and that provides it the invasiveness after L-selectin troponin is activated, and they attach to their ligands, and then it will invade into the epithelial lining of the endometrium and will try to reach down into the connective tissue, the stromal cells present at the, or stromal cells of the endometrial lining of the uterus, right? That is what they try to achieve. So the next part of the implantation, okay, and the menstrual cycle in the negative and positive feedback loop manner, okay, will be taught in the next class. And if you have any doubt, you can ask me on my personal number. So this is a very conceptual chapter, okay. Adhere to every concept in order to solve any kind of comparative level questions. And even if the questions are quite tough in any kind of examination, board examination, NEET examination, it's the concepts that will get you through. Okay then. Thanks a lot. Let's meet you in the next class.